Hello, my name is Ishani. I'm going to be a senior in the fall at Cranbrook, and I'm one of this year's Discovery to Cure interns.
Good morning. My name is Myrna Stephan. I'm also a 12th grade student at International Academy, and Ishani and I are partners in the Pilsner Lab, where we amplified the mitochondrial genome for sequencing to discover mutations and deletions. We also investigated the phthalate association and sperm epigenetic age in the seeds cohort.
Our first project aimed to optimize the polymerase chain reaction, or PCR, to amplify the human mitochondrial genome for sequencing to discover mutations and deletions. Mitochondrial DNA carries 16,569 base pairs, with 37 genes that encode for 13 proteins. All 13 are related to the production of proteins involved in cellular respiration.
While the structure of mitochondrial DNA is consistent across the various different cells of the body, the amount of mitochondrial DNA is not. mtDNA copy number is a measure of the levels of mitochondrial DNA that exist within each cell. This table shows how the mtDNA copy number varies across the different cells in the body. And of these, the male germ cells has the lowest mtDNA copy number, making it a challenge to amplify the mitochondrial genome, to amplify the sperm DNA. Our project focused on optimizing the PCR to amplify the sperm mitochondrial genome for sequencing to discover mutations and deletions.
In trying to optimize mtDNA concentration, we experimented with two different primer sets, which gave us two products, Amplicon-1 and Amplicon-2. The forward and reverse sequences for both primers can be seen on the slide. The forward primer for Amplicon-1 binds to the M2-RN1 gene, shown in purple, and the Amplicon-2 forward primer binds to the MT-ATP6 gene, shown in red. Primer set 1 produces a product of 8,397 base pairs, and primer set 2 of 8,289 base pairs. They are both long sequences, so we had to find a master mix that is recommended for long PCR product. The primer sequences were taken from a publication by .NET.
We had to optimize various different variables within this project. For example, we experimented with two different master mixes, the QIAGEN All-TAC master mix and the Takara LA-TAC master mix, which was used in the previously mentioned publication. Other variables we experimented with included the annealing and extension time and temperature, the number of cycles, as well as the template DNA concentration. These variables changed because we used protocols from both the publication as well as the protocols that came with the master mix kits. We amplified the PCR using Ishani and I's genomic DNA, which we extracted in the lab, as well as the sperm DNA.
We ran a couple gels every week, but have chosen to just highlight a few in our presentation. The gel here shows our attempt to optimize annealing temperature for the amplicons by running a temperature gradient that ran from 55 to 65 degrees. These images show the results. The gel on the left shows the gradient for Amplicon-1, and the gel on the right shows the gradient for Amplicon-2. You can see that Amplicon-2 yielded much cleaner results. You can clearly see the effect of the temperature gradient. From these gels, we concluded that neither amplicon performed well at lower temperatures. In the 55-degree band, you can see non-specific product. We decided to choose a temperature in the middle as a way to accommodate for both primers. So we moved forward with an annealing temperature of around 60 degrees.
These gels show the result from our different master mixes. The gel on the left was our first attempt with the QIAGEN master mix, which was Ishani and I's somatic DNA. We can see bands in every route, however, Amplicon-2 performed significantly better. Next, we experimented with the Takara master mix. These are very faint bands for a few of the vowels, however, the brighter ones were the samples from Amplicon-2, which are consistent with the results from the QIAGEN master mix. The Takara master mix did not produce consistent, clear results. So therefore, we decided to continue with the QIAGEN master mix. However, you can see the results were inconsistent, as the somatic DNA bands look much clearer in the first attempt. We also tried to amplify sperm DNA in this gel, but we were unsuccessful.
Some key findings we established from this project was that an annealing temperature of 60+ degrees works best. Additionally, a template DNA concentration for sperm is best at 5+ nanograms. We also saw that while the QIAGEN master mix produced more product than the Takara master mix, there was much more non-specific product. Therefore, we can conclude that our method requires further optimization before sequencing will be possible.
Our second project looks at the urinary phthalate metabolites association with sperm epigenetic age, or C, in the seeds cohort.
Before we explain our project, we'll give some background information on male infertility and how it's measured. Infertility is a disease of the male or female reproductive system defined by the failure to achieve a pregnancy after 12 months or more of regular unprotected sexual intercourse. There are four main causes of male infertility. 1, low sperm production. 2, abnormal sperm function or blockages that prevent delivery of sperm. 3, illness and injuries. And 4, lifestyle and environmental factors.
Male fertility is assessed with 5 semen parameters: semen volume, sperm concentration, sperm count, motility, and morphology. These parameters are assessed by trained clinicians at IVF clinics. An individual is classified as infertile using the reference levels set by the World Health Organization, or WHO. For example, if one's semen volume was only 1.2 million per milliliter, he would be considered infertile. However, these parameters are poor predictors of pregnancy outcomes. So we need to find other biomarkers to successfully predict pregnancy outcomes. A few years ago, people started looking at epigenetics because it was shown that environmental factors may affect not only your gene sequence, but your gene expression as well.
So as Myrna said, studies have recently shown that environmental exposures can impact the development of your genes and how easily they turn on and off. DNA methylation is one of several epigenetic mechanisms that cells use to control gene expression. DNA methylation does not change the gene sequence. However, it is the process in which a methyl group is added or removed from a gene, causing an increase or decrease in regulation of gene expression.
Epigenetics can also affect the rate at which your body ages. Everyone has a chronological age, which is just how many years it's been since you were born. However, every cell in your body ages at a different rate, some faster than your chronological age and some slower. The sperm epigenetic age is how old one's sperm cells are due to the environmental factors that during their lifetime. C is calculated using machine learning software called SuperLearner, which takes a sperm DNA population from 790,000 methylation sites in the human genome, as well as a man's chronological age, and gets an output of a predicted age, which we call C, used based on the methylation patterns.
One of the environmental exposures studied in the Pilsner lab are phthalates. Phthalates are often found in plastics to make them more durable. They're also found in cosmetic products like nail polishes and perfumes to serve as stabilizers and solvents. Multiple studies have proven the negative effect of phthalates. For example, prenatal exposure can affect neurodevelopmental outcomes. Additionally, exposure during pregnancy can impair brain development and increase risk of learning and attention disorders. Studies have also found that phthalates can cause harm to the male reproductive system.
All 15 phthalates that we studied belong to one of 3 groups: high molecular weight, low molecular weight, and phthalate alternatives. The high molecular weight phthalates are often found in plastics, food packaging, and vinyl products. The low molecular weight phthalates are often found in non-plastic applications. Each phthalate quickly metabolizes into their metabolites, which are found on this table. These metabolites are the compounds which we studied.
So the aim of our second project is to investigate the association of exposure of phthalates and their mixtures with sperm epigenetic age in seeds participants.
We are working with a human cohort SEEDS, which stands for Sperm Environmental Epigenetics and Development Study, which was done in Springfield, Massachusetts. SEEDS includes almost 200 couples. Our project focused on the information about age, BMI, and race of the male participants. The SEEDS data also includes smoking status, which was confirmed by looking at the cotinine levels within the urine samples. Urine-specific gravity looked at how concentrated the urine sample was, which is determined by the number of proteins as well as various other variables. Batch refers to the 3 batches that were used to analyze the urine samples. Each batch used a different data collection method, so we kept track of the batch number for this reason. The SEEDS data also includes C, which was calculated based on the sperm DNA methylation. The urinary phthalate metabolite concentration was also included in the data and measured by the CDC.
All our calculations and graphs were generated in R and include 173 male participants. The first parameter we looked at was age. The bar graph shows the range of ages within our dataset and how age was distributed across each category. As you can see, the 30 to 40-year age category had the most observations, and the median age across all categories was 36 years old. The figure on the right also shows the distribution in age within the three categories. Any outliers are also visible.
On the left is a bar graph, which shows the number of observations within each category of BMI. The parameters for categories in BMI came from the WHO. Here, the majority of our participants were classified as overweight. On the right is a box and whisker plot, which shows the BMI distribution between each category. The median here was about 25.
In a similar manner to BMI and age, we calculated the mean and standard deviation of urine-specific gravity. We will eventually use the specific gravity measurements to normalize the phthalate metabolite measures. Additionally, we looked at the number of currently smoking participants, and the race pie chart shows the distribution of race within our participants. The data shows that a majority of the participants in the study are non-smoking and non-Hispanic white.
Our next step was to analyze C. This graph shows how we characterize C as well as the distribution. We considered those whose C was less than -1 as younger, -1 to 1 as the same age, and greater than 1 as older than their chronological age. There seems to be an even distribution between the three categories of C. We calculated a mean value of -1.73.
All of the urine samples were sent to the CDC where they calculated the phthalate concentration. However, the machines that were used were programmed with a limit of detection unique to each phthalate. So we created a graph calculating what percent of the data had a phthalate concentration value above the limit of detection. The phthalates all had over 50% of observations, except for MCOCH. You can even see that the majority of the phthalates have 98 to 100% of observations greater than the limit of detection. The rest of the columns display the mean, median, 25th percentile, and 75th percentile phthalate concentrations.
Here is the same table, except at this step, we normalized the data according to the urine-specific gravity, which looked at how concentrated the urine sample was, determined by the urinary dilution. We normalized the data according to the urine-specific gravity to compare the phthalate levels, and we used this formula to adjust the new phthalate concentration levels.
Next, we used the raw data from the previous slide to create histograms for each phthalate. Alongside the raw data, we also made log and log-based transformations for each phthalate concentration. We used the raw data as well as both transformations to create histograms for each phthalate. We were looking for the transformation that best resembled the normal distribution curve. Looking at the phthalate MEHHP as an example, we can see that the log-based 10 transformation created a distribution that most resembled the normal distribution curve. This trend was manifested for the rest of the phthalates, and so we used the newly log-based 10 transformed data for the following steps of our project. Unfortunately, we did have one outlier. The MCOCH phthalate did not follow the same trend.
You can see here that we were unable to achieve a normal distribution with any of the transformations for MCOCH. We believe that this was because most of the values for MCOCH were below the limit of detection as we saw in the previous table. Therefore, moving forward, we set a different adjustment for this phthalate. We assigned each value either 0 or 1, depending on whether it was above or below the limit of detection.
Next, we created a figure using the Spearman method to check the correlation between different phthalates using Spearman's rho. If the number within the circles are close to -1 or 1, the phthalates are thought to have a strong correlation. This means that both phthalates are often found together in a sample. For example, the top 4 compounds display large rho values, which is expected because they share the same parent compound, DEHP.
Next, we created a figure using the Spearman method to check the correlation between C and the 3 covariates. A covariate is a measure that controls for anything that may have an effect on both the exposure and the outcome. In this case, we use BMI, age, race, smoking status, batch, and urine-specific gravity. Of these, the smoking status was the only covariate that suggests an effect on C. It was shown that those who did smoke have a higher C. However, there were only 14 participants who smoked, and so the data may be slightly skewed.
Next, we created figures using the Spearman method to check the correlation between C and the log base 10 transformed phthalates. None of the results were considered statistically significant as the p-values were not below 0.05. Therefore, we can conclude that none of the phthalates are correlated with C.
We also created regression models to check the correlation in a different way, beginning with the covariates. You can see here the smoking status suggests that there may be a correlation between smoking and the sperm epigenetic age.
We also created two different types of C phthalate regression models. The unadjusted model is a regression between C and the phthalates adjusted only by urine-specific gravity. The adjusted model is a regression between C and the phthalates adjusted by all 6 covariates.
Here is the results of the unadjusted regression model. The graph on the right is a visual representation of the table. The point estimates are represented as the red dots on the graph. The 95% confidence intervals are shown as the ends of the black lines. Here, we can see that none of the phthalates are associated with C.
We made the same table and graph for the adjusted model, which checked the association between C and the phthalates while adjusting for all 6 covariates. We did not see any p-values that suggested significance, and our plot did not show any of the phthalates' confidence intervals lying completely above or below the dotted line. The results are consistent with the unadjusted model shown in the previous slide.
However, as humans, we are not exposed to only one phthalate during our lifetime. In order to create a more accurate representation of human interactions, we created a mixture model, which allows us to look at the effect of multiple phthalates on C at one time, which is something that the regression model did not let us do. Using the mixture approach, we were able to find the PIP value for each phthalate. PIP stands for the posterior inclusion probability, and it is a ranking to measure how much the data favors the importance of a phthalate on C. The closer the PIP is to 1, the more likely that it is for the phthalate to have an effect on C. From the mixture model, we can see that none of the PIP values are distinctively high, which suggests that none of the phthalates have a strong association with C. The graph on the right shows the overall compound effect between the phthalates mixture and C. Here we can see that there seems to be a dose response which diminishes at 60%. Unlike our previous findings with the regression models, the mixture model suggests that there may be a correlation between the lower percentile phthalate concentration levels and sperm epigenetic age.
In our last step, we divided our phthalates into 3 groups, which were high molecular weight, low molecular weight, and phthalate alternatives. We then checked which group was most associated with C. The PIP values for each group all hover at around 0.63, which suggests that each group has the same effect on C.
Some key findings we established from this study is that smoking was significantly correlated and associated with C in the seeds cohort. Phthalate metabolites did not correlate with C. Urinary phthalate metabolite concentration does not show a significant association with C. And the BK-MR mixture model suggests a dose response between the phthalate metabolites mixtures and C. However, this trend was not consistent across upper quintiles.
In conclusion, the individual model did not show an association between phthalate metabolites and sperm epigenetic age, but the joined models showed a dose response of association, suggesting that at lower quartiles, phthalate exposure increases C.
We'd also like to thank a few people who made this day happen. We'd like to thank Dr. Carolina Nowak, who could not be here today but played a vital role in our learning throughout the past 8 weeks. We'd also like to thank Rona Wang for teaching us and guiding us through all the bench work. We'd also like to thank Dr. Pilzer and Dr. Moore for giving us opportunity to work at the lab in the CS Mott Center. Thank you to everyone else in the Pilzer lab who spent time teaching us over the past 8 weeks. We appreciate you.
Very nice. If somebody has a question, feel free. The students also can ask questions. Okay. If no questions, Elisa will move to the next one. Hi, Joe.